So on behalf of my co-organizers, Geoff Wahl and Brian James, thanks very much for joining this latest installment of the Space Force meeting. Today's speaker is Amjad Askary, a postdoctoral fellow in Michael Elowitz's lab at Caltech. Amjad has developed a really cool approach for multiplex in situ imaging of combinatorial barcoded cells in situ. And this is suitable for spatial cloning, clonal tracing, and lineage reconstruction of cells in tissues. He published this in a very nice paper in Nature Biotechnology last year, a technology which he called Zombie. I'll let him explain where the name comes from. And I think most recently he's been applying this to studies in mouse retina, which is obviously a highly organized multicellular tissue, and so I think very appropriate for studies such as this. But I think also this technology is going to be very applicable to other aspects of development in disease states such as cancer and other degenerative conditions associated with aging. I think a really exciting technology. So we're really happy that Amjad has taken this time out of his schedule to speak to us today. So I just want to say, if you could use chat for questions, and then at the end I'll either unmute people or read the questions out myself. So with that, I will hand over to Amjad. Thanks very much, Amjad.
Okay, thank you for this nice introduction. Thanks for having me here, and thanks everybody for taking time and attending the presentation. So first about format, I was wondering if I'm gonna just talk and then ask questions at the end, or if usually like people interrupting, it's fine with me. But if people are gonna ask questions, I think it's good to be interactive. So I need to keep an eye on who's raising hands. So sure way.
Yeah, whatever you're most comfortable with, Amjad. If you want to take questions as you go through, that's fine. Otherwise, we can save them to the end. I've been told I need to mute my microphone. There's a bit of an echo coming through. So likewise, if everybody could mute if you're not already muted. Thanks.
Okay, so feel free to interrupt if you have questions. I think that would be more fun, probably. So I'm very excited to talk with you guys today about the work I've been doing in Elowitz in the last few years, and also the direction that we're taking it to study the developmental ability.
Sorry, Amjad, there is still a bit of an echo coming through. I don't know whether that's because people are not muted or whether it's something with your microphone.
Sorry. Let me try to. I can. Switch the microphone. Is this better now?
That may be better, yeah. Let's try again.
Okay. Yeah, sorry for that. So, so the technology that we want to talk today mostly is about barcoding, and barcoding has a lot of applications in biology and biotechnology in general. So overall, wherever you want to follow a group of cells over time, barcoding helps you to do it more efficiently. For example, if you can label cells with inheritable barcodes, you can use those to study the clonal relationship between the cells. Or if you want to do multiplex screening, it helps if you can encode each condition, in this case, expression of the gRNA, with a barcode, and that helps you to mix the cells, mix many different conditions together, and be able to decode that at the end of the experiment. More recently, a new type of barcoding has attracted a lot of attention, where the barcodes, instead of being static labels, are actually dynamic.
Sorry, Amjad, one more. If you, I'm told if you could maybe move it a little bit closer to the microphone, that might be a bit clearer.
Oh, okay. Let me.
But it's I mean it's audible to me. It's not the clearest, but it is audible. But if you can move a little bit closer, that might help.
Yeah.
Do you want to give it another go?
Should I start over, or was it?
That sounds, when you just said that sounds much better.
Okay.
You can continue where you are. You don't need to start over.
Okay. Yeah, I'm very close now. It's a good thing that the Zoom is such a small thing. I'm thinking at the top. Anyway, so in all of different barcode methods, you need to be able to read out because at the endpoint of the experiment, obviously, and there are different modes that is. The one that is more dominant at the moment that the method people use is through sequencing. So I'm showing here on the left. And this is a great technology because it's scalable, it's relatively easy to use, and because it has a single base pair resolution, you can have varied libraries that can label a lot of different conditions or a lot of different cells uniquely, and then you can dissociate the tissue and see and recover this information. However, it does have this fundamental limitation that you need to dissociate the tissue to be able to run the sequencing reaction. And because of that, you lose a lot of the spatial information that in a lot of contexts, especially in the nervous system, is very cool. So there are alternative methods showing one example of it on the right. Where you use microscopy to code genetically inherited barcodes. The good thing here is that because you're using microscopy, you have the morphology of the cells, organization of the tissue, and all spatial information. And also because you're using microscopy, you're losing your cells in the process of preparing sample. But at the same time, there are much fewer combinations of barcodes that you can have. In this case, I'm kind of using barcode very loosely. And you have three fluorescent proteins. So the combination of these three fluorescent proteins can only give a handful of different combinations. So each of these methods has advantages. There's basically a trade-off between the diversity, the labels that you can have, and the spatial information that you can make. So ideally, what you want to have is something that you can maintain the spatial information, but also have a high diversity of barcode. That's why we thought the imaging of DNA barcodes in situ can be a solution to this problem. You can have an arbitrary number of combinations for DNA sequences, and if you can image them by microscopy, that basically gives you the advantage of both of these methods. And fortunately, over the past few years, there has been a lot of advances field of multiplex imaging, spatial transcriptomics. You're going to hear in this seminar series about. And in fact, it was chosen as a year in 2020 by Nature. So I'm not going to introduce all of the different technology being developed in this field. I'm going to just focus on that we happen to use, and that is on sequential hybridization and imaging. So this category of technologies, they basically work based on hybridization, set of fluorescently labeled probes. An example here, purple. And you hybridize this probe to your target, you image a few channels, and you dissociate them with different methods, and then you repeat the cycle over and over. And each round, if there are enough fluorescently labeled probes hybridize your target, you can visualize it in the images as one of these diffraction limited dots that you can see in the picture below. So I'm showing one example here. Color of these dots that are associated with individual RNA molecules in this case can change from cycle to cycle based on how you design your probe set. So basically, you can have temporal code each RNA molecule. So in this case, for what is called mRNA1, this is one molecule shown in yellow in the first round, and then cyan yellow again. So this is temporal code for mRNA1. And the important thing here is that this code book grows exponentially with the number of rounds you do. And leveraging this exponential growth plus some automation, you can actually scale this up to the transcriptome level, and you can image the entire transcriptome using this approach called seqFish. So this method really well for imaging mRNA in the cell, but it still has fundamental limitations when it comes to imaging barcodes. And the source of this limitation comes back that for you to be able to see a molecule as a dot that I showed, there need to be multiple probes binding to it. Each one of these probes is a fraction of the event signal, and by themselves, one of is not enough to have a reliable signal. Because you need to have multiple of these probes, depending on the use, somewhere between 20 to 40 of these hybridized to your target, the target needs to be large enough to read them. And this is usually not a problem for an mRNA, but when it comes to barcodes, this is a problem because for accommodating 20 to 40 probes, your barcode needs to be 500 to 1 kb large. And this is the unit of information for barcode. So to, for example, sequencing, you have one base pair being the limit of detection. So if you have barcodes that are 100 to 1 kb large in each unit of them, then you want to make arrays of them. You're going to deal with a very large and sequence that is hard to introduce to cells, and also hard to express. This expression actually the second problem. Information is encoded in DNA in the systems, but for us to be able to detect it efficiently, we usually need to convert RNA by transcription. And this transcription is not always efficient in different cell types, especially if you're dealing with a large construct. So these were the problems that we were hoping to solve. Basically, ideally, we wanted to have a method that allows you to detect barcodes in situ very efficiently, ideally with one probe, and independent of endogenous expression of these barcodes in the cell. And the solution that we came up with was using phage RNA polymerases. These are robust RNA polymerase enzymes that can identify a very specific promoter sequence and then transcribe it downstream of that sequence. And the idea here is that you engineer your barcodes downstream of one of these promoters, for example, T7. And because the promoter is not present in living cells, these barcodes are not expressed, but they are available for editing. For example, if you want to mutate them or do that. And then after the experiment is done, fix the cells and induce the RNA polymerase exogenously, which will then find the promoter, transcribe the barcodes. And under the right condition, the idea is that you can trap or fix a lot of these transcripts in the active site of transcription. And by immobilizing the transcript in the active site, you effectively amplify your signal, and that's how you to detect the smaller regions, as showing in the future slide. So the basic features of, by the way, we call it zombie because it's basically reviving the dead barcodes in the cells that are already fixed. And the features of this barcode readout method I emphasize is that it's independent of endogenous expression, because all the action is happening after the cells are fixed. And I show during the presentation, it enables us to detect short bonds or short sequences of DNA, short as 20 base pair, and also discriminate between them based on single nucleotide variations. So to test this method or to develop this protocol, the first thing that I did was making a mouse ES that has one fishable sequence, one sequence can detect with fish, downstream 3 of these phage promoters. There's T3, T, and SB6 were the ones that I used. And we basically our protocol by which we can, for each of these polymerases, we can transcribe the barcodes after the are fixed. So you can see here both the and the polymerase need to be present for transcription to occur. And you can see the transcription active sites as this bright spots where RNA is accumulated. So we can detect these sequences in situ without them being transcribed in the living cells. But the next capability I'm excited to test whether or not this method can enable us to detect shorter DNA sequence. And the idea is that in the active site, you have multiple transcripts of the sequence of DNA. So there's going to be multiple bind sites for each sequence of probe. For example, the red probe is designed against this red piece of DNA, and there's multiple copies of the active site. So the probe can bind to each one of them, and now different probes are brought together. So as opposed to using the transcript to bring together multiple different probes, you're using the abundant transcripts in the active side to achieve the amplification or the signal. The same for the green and yellow region. And this is what is said here. You do for the three page promoters or polymerases that we use. Each panel shows a 20 base per region that is probed with one individual probe, and you have the red, green, and yellow signal being very bright and clear, and also overlapping with each other. That kind of assures you that you're looking at the specific signal and not background noise. So this was very exciting for us because by reducing minimum detectable size from something like 500 base pair or 1 kb to a 20 base pair sequence, that means that you can relatively easily concatenate these 20 base pair sequences and make combinatorial barcode libraries. And this is the first example of such libraries that we made. It's a proof of principle. So we went for a library that had 4 variable positions. And each one of these variable positions can take one of 3 variants. So they're shown as A, C, 1A, 1B, and 1C in this position. So basically, this gives you 81 different combinations with 12 different sequences that you need to probe for. So what I want to emphasize here is that the number of combination is going to grow exponentially again with the number of variant sites or positions. So as you can see on, we can scale this up without increased number of sequences that need to be probed proportionally. But with basically low diversity library, what I want to show you is the basis of this method. So in this picture, we packaged the barcode library in a lentivirus, introduced it to developing cheek areas to label the neural tube cells. Shown here, each panel is corresponding to one position in the library, and I'm showing 2 clones. One is shown by circle and one square, and the colors correspond to the variance in each of the positions. So, for example, for the circle clone, in position 1, you have variant 1 shown by cyan. In position 2, you have variant 2 by magenta, and so on. So when you basically convert this 1 to on base 3 to decimal, you get the variant number 11 or combination 11 and labeling clones, and combination 6 labeling the square clone. And we use this library to basically, as a proof of principle, trace clones in the chick neural tube and also in the chick red. And also we compared the frequency of barcodes that we're imaging with sequence to kind of make sure, as an independent validation, make sure that what we're seeing is actually real. And after we decided that this method is actually working, we spent time in scaling up in both developing protocols compatible with automation, so you can do it not manually, and also screening a lot of sequences to find barcode sequences that we can teach you robustly, and also coming up with a schemes robust to error in the readout. So even if you have one or calls in the readout or a failure to detect the readout, we can still decode the library faithfully. And with all of these different developments together, we were able to finally make libraries that have tons of different combinations, and even more than 1 million different combinations that are now on the scale relatively comparable with the sequencing-based methods for readout. And what I'm showing here on the bottom is actually just the distribution combination indices in the libraries. So what I want to emphasize here is that even there are some barcodes in the library that have higher frequency errors, overall library is not dominated with the few combination have like orders of magnitudes higher higher levels, and it's relatively uniform. And also we showed that we can, in fact, with using the principles that I showed for low diversity library, we can actually read out this higher diversity libraries as well. So here what I'm showing is cells that are infected with high density of infection with one of the libraries, and right, I'm focusing on one of these integration sites. And each round of integration, again, you're looking at one of the functions in the library, and all can put these numbers together, code with which combination is present this side. Putting all of these together, we basically have high diversity viral libraries that we can use to label cells, and then we can detect this by imaging endpoint. This is very exciting. There are very different possibilities that you can take. The one I'm mostly excited about is using it to study variability in development, specifically in the retina. So one of the intriguing aspects of development for me is that it's capable of producing very robust and reproducible outcomes, even though many of the processes that are involved in it are very variable and sometimes even inherently stochastic. So for example, if you look at the clone composition from individual, it's different for the same tissue. Or if you look within the clones, lineage trees that make up those clones are different from clone to clone. But at the end, you get a very robust and reproducible structure most of the time. And this is kind of exemplified case of retina, very unique structure with a stereotypical cellular organization. It is made of 6 major neural cell type and one type of glia that are organized together in this layers of cells. In the outer layer, you have receptors that capture light and convert them to electrical signals, and then there is an nuclear layer where this signal is processed. I'm sorry, do you?
Sorry, Amjad.
Are you still there or lost?
Yeah, but I think we've lost your screen. Can you share again?
Let me share it again.
Okay, that's good.
Okay, yeah, okay, good. So you see that this organization is actually very important for the function of the tissue, and needs to be maintained for the tissue to be able to function properly. But when you look at how this structure is built during development, you see that it's actually made of very variable clones. And what I mean by that is that if you label an individual progenitor cell in the retina early on in development, and then come back later and see how many cells it gives rise to and what cells it give rise to, you have a wide distribution. It can be anywhere between 1 or 2 cells all the way to more than 200 cells. And clones can be made of different combinations of cells, pretty much any of cells that you can think of. And more pleasantly, at least to me, is that these ones do not intermix much either. So each one is radially packed together, and there's not a lot of tangential distance between the clones. So when you actually put these facts together, you have basically a very organized structure that is made from various parts. And the question is how that can happen. So one way that this could be happening is through extrinsic communication between the clones. So for example, you can imagine that one has a lot of an individual cell type, and then it kits it somehow with some signal to others to not make as much of the same cell type. There are actually candidates for this type of interactions happening. For example, sonic hedgehog is shown by genetic manipulations that is made by gamma cells, and it is inhibiting production of Ferdinand cells. But also in some other model organism, in zebrafish, where the time-lapse imaging is people have seen that the composition of each individual clone, you can fit it basically as a stochastic model. It doesn't seem to have a lot of from surrounding clones. And you can also imagine that in mouse, maybe there is a condition where stochastic divisions of cells produce scenarios in different individuals, then get resolved by these extrinsic signaling layers. So what I want to argue here is that to be able to actually understand this system, we need to take account of variability from individual to individual, and we need to be able to label a lot of clones in each sample to be able to look at the interactions between them and quantify the stochastic effects. And this is the direction that we're taking with this libraries. The first thing we did was to just use empirical distributions that I showed in a few slides back to make sure that we have enough diversity in the libraries to be able to move forward with this project. And what I'm showing basically using the libraries that we have, we can have up to 300 clones or intermingled or just adjacent to each other, but it's able to reliably classify clones in more than 95% of the cases. So not any sort of collision problem as a result of cells from different clones being next to each other. So you don't really need to rely on any physical boundary between the clones to be able to identify them using these. And then we just what I'm trying to show here is basically that the fact can introduce this bar either through transduction or electroporation into the now, and then detect. I'm just showing here a couple examples of like 2 barcodes being detected here, but you can with the same principle I showed earlier, basically do this sequence. And another component of being able to classify as a clone is that in addition to barcodes that tells you a clonal history of the cells, you also need to be able to identify the cell types. And for that we basically choose a set of genes that mark different major cell types in the retina. And what I'm showing here, multiplex in situ procedure that allows us to make these different genes, either most of them actually with RNA imaging, but some of them with antibody in the same sample, so that you can have basically labels for all the clones. And to all of these technologies is going to allow us to basically trace the clones at high density in the retina. And I just wanted to show a glimpse of like some preliminary analysis here of the things that that we're interested in doing in this project. Here, using some data on the clonal tracing, we basically did a dimensionality reduction to show that there's already some structure in the clonal composition of the retina. You can see clusters of the different clones. So here, what's distinguishing between these different clones is the number of cells that are in the cell types that are made of. And when you basically look at what drives clustering, some of the correlations that you see can be explained as known in the literature. For example, the cone and horizontal cells are correlated, and we know there are some fate restricted printers in the retina that produces these 2 cell types. rods and bipolar cells are correlated. Again, these are the cell types born later in the retina, so it makes sense for them to be correlated. But you also find some correlations that you could not predict or expect from the literature. For example, this correlation between molar glia and amacrine cells together. So these kind of unexpected correlations out of these studies is what I'm excited to follow up on with functional studies later on. So everything that I showed so far in the presentation has to do with type of barcoding where you have a label in the cell that kind of is fixed. You introduce it at one point, and then it stays the same way all the way. The application that I'm interested in is tracing, but you can also use the same label in the multiplex screening procedure. But I want to spend the second part of the presentation kind of talking about this other type of barcoding that is what we call dynamic barcoding. So the barcodes change over time and modified. This is a more recent development is enabled by engineering technologies that have been developed in the past decade, and it's very exciting time for these type of technologies. So the way I would like to think about these overall is kind of an analogous to memory in the computer. So you have a barcode that is initially in one state, like let's say it's this, and then it's modified that usually this modification is irreversible. So you go from one state to another, and this modification can happen by a variety of different technologies. It can be with CRISPR-Cas9, or it can be with integrases or bases or beta. There's different ways of engineering the genome. And if you have an array of these barcodes, all, for example, in one state, and then stochastically mutations or edits happen array over time, then you can use the state of this barcode arrays at the point to kind of infer the lineage relationship between this, because the relations are inheritable and also irreversible. The principles that are used in this lineage reconstruction approach basically similar to phylogenetic tree reconstruction in evolution, where you have mutations between different species. Here you have mutations between different cells that are kind of accelerated using those genome technologies. Further, if you link the rate at which these mutations occur to some sort of signal. For example, if you have the gRNA that initiates this edits controlled by a signal responsive promoter, then you can have a read in the cells of how much signal the cell has perceived. So the higher the signal, the higher the gRNA and the edits. So you're going to have more edits in the cell. And at least based on simulation, you can say if you have this edits accumulating parallel channel in a different set of barcodes, than the ones that you use for recorrecting the lineage, then you can not only construct the intensive signal in cell, but also when they happen, have a scaffold of lineage tree that you can basically with this signaling event that are in a different barcode channel. So this is a very exciting opportunity, and a lot of different groups got excited about it. So there's a healthy diversity of techniques and approach for achieving the same result that are particularly published in the last few years. Our lab is specifically had this vision from the beginning to be able to actually look at a tissue section and be able to not only see the final state of these cells, which is where they are in the tissue, but what genes, what type of cell they are, but also kind of reconstruct the lineage relationship between them and reconstruct the history of the gene expression in them. So that was kind of the vision of a subgroup within the Elowitz group called Memoir, that to use this genome engineering technology together with the image-based barcoding for achieving this ability. And the first version of this recorded a few years ago used this specific type of biological memory called barcoded scratch pads. So these are made of two parts, each one of them, and there is a pad, which is what is targeted by Cas9, and there's a barcode which makes this a scratch unique. So the barcode for different integrations of a scratch pad unique. And then when this barcode scratch pad is expressed, because it has these two components transcribed on the same mRNA, you visualize it by a single pushing the cells as two dots with two different colors, one associated with the scratch pad and one associated with the barcode. So in this, here I'm showing two cells. In the upper cell, you have a lot of red dots that have associated green dots. So that means that you have a barcode and a scratch pad. And then when the Cas actually cuts the scratch pad and causes a collapse on it, then you only have the signal associated with the barcode and none with the scratch pad. So only green, not red next to it, shows the collapsed state. This is just a way of distinguishing between the 0 and 1, as stated earlier, with a single molecule fish. And the actual system was made of multiple integrations of these barcoded scratch pads, areas in the different chromosomes of the cell, and then two other components that you need for cutting them. One is Cas9 that was expressed ubiquitously by a PG motor, and another one is gRNA that was controlled with the Wnt reporter. And when you have this multiple different integrations of barcoded scratch pads, and you have the Cas9 gRNA activated, then you accumulate these stochastic edits as the cell grow, and using the same principle that I just mentioned, you can reconstruct this relationship between the cells. So that was the first implement of the memoir system. And it was a good proof of principle, but it had some fundamental issues that limit its utility. Mainly, one of them was that the scratch pads or the barcodes, it was hard to have them ubiquitously expressed in the cells, especially because they were scattered in different chromosomes. It was hard to have a consistent expression of them in different cell types. Also, because you could not really put these in an array of barcodes. You had to have them each individually in a different place, because they've been targeted by Cas9, and because they need double-strand DNA break to work. If you put multiple of them in one array, there's going to be collapse at the array that removes a lot of information that is already recorded. So if you have 10 of them together, you can have the first one and the eighth one basically getting caught, fused together, and you lose the information in between. So these were basically drawbacks that we thought that we can solve with this new in-situ readout technology. And the way that went about them was just to use not just CRISPR-Cas9, but a better to make writing of the information in the barcode. So this editor, if you're not familiar with them, these are a newer addition to the CRISPR toolbox. They're basically made of a mutated Cas9 fused to deaminase, and they do not make double-strand breaking DNA, but can target a specific base change from, for example, A to G in a specific region into the genome. So basically perfect tools for writing information in the genome, because they're programmable and predictable, but at the same time, they don't make double-strand breaks. They don't have the same problem that we had the first version of memoir. Double-strand break prohibited us from putting a lot of these barcode array. And by the way, the reason we need to put them in an array is because we need a lot of these individual units to be able to the lineage. Just a few of them is not going to work. A few information is not enough. We need the scale of 100 of them to be able to reconstruct lineage trees of significant depth. But the question is, can we actually detect this individual base pair changes with imaging? And the idea here was that, again, using the same zombie amplification technology, we have an active site that has multiple copies of barcode, and we compete against each other probes or two probes that are identical with each other, except for one base pair in position. And the one that is a perfect match for the target is going to more target size and active size. The rest of them are mostly washed away, and this different in binding affinity is going to translate in intensity, which then we can quantify to call visual base pair in this position. So this is kind of experimentally shown here. Here we have a sequence that has A in this region that we're interested in. So the target is going to A. The four pros that are coming against it. One of them has a T, which is a target match, and the other ones have one match here. And you can on the right, I'm showing 4 cells, 4 different experiments, basically the same in conditions, and in each condition you see probe that is associated with T base pair as the majority of the signal. So if you look at a column, regardless of which channel the probe it's in, it has the higher signal intensity. So this is shown here for one individual cell as an example. It's also quantified here over many different cells. So you can see very reliably you can basically call the right base pair here. And this was for cell culture. We wanted to also see if we can do the same thing in vivo, because in the context of the two, everything is more complicated. And for doing that, we packaged this barcode lentivirus, and we introduced it in both chicken embryos and mouse samples, and showed that first of all, qualitatively you see the same effect. Again, the match probe has the dominant C compared to the mismatch, regardless of what channel the mismatch is in both different animal tissues that listed. And also we quantified it by making these lentiviral pairs where you have 2 of the same base pairs, for example, G and G on one virus and A and an A on another virus. So for each integration site, you have to detect either both G, and if you detect an AG or GA, that's considered a mismatch, considered a failure of the gene. And this enables us to quantify this in vivo in the tissue samples, so that we have about on average between the different samples that we tried, an average about 92% accuracy for calling this into base pair changes in vivo. It's the higher in the cell culture. So when kind of confident about our ability to detect these base pairs, then I designed this memory arrays, or these bits that have one single byte A that can be converted using base editors. And also each one of them has a tag or a barcode ID next to it that makes it unique. So now you have an array of, for example, 12 or 18 in different cases of these barcodes, each one of them one single nucleotide that can be converted from A to G using the base editor, and each one of them can be clearly identified in situ. And we have 2 designs for these. One design where different gRNAs target different bark. This is good when you want to have multiple parallel channel recordings, so each channel being encoded with a different gRNA. And another design where a single gRNA can target all of the barcodes in an array. The part that is being targeted is the same. The difference between them is only based on that tag sequence. In both cases, you can see visually pretty straightforward to distinguish the edited and unedited barcodes. Also, quantitatively, you can cluster them based on the edited channel and unedited channel. And as you expect, you only get the edits when both the base editor and the RNA are present. And again, I'm showing here this for one barcode in each system. You can do the same thing for all the other barcodes as well. And the edit rates that you get is consistent with sequence, what you expect to see. Also, the clustering that we do here is robust to bootstrapping. So all of this together basically makes us confident that we can have arrays of 12 barcodes that we can edit it using base editor. We can reliably detect in situ. And these are now the ones that we use to perform the new version of memoir experiments. So the direction that I'm taking with that is that I made a lentiviral library each copy of lentiviral genome has two of these together. They make up 36 bits of information, and each one has a unique ID barcode. Each integration into the genome can be identified on this ID genome ID barcode. It also encoded the RNA for one of these two arrays. And we're now in the making of transgenic mouse line using this lentiviral library that we can use and for initiating the required coordinating with a base editor mouse at different stages. But because this transgenic mouse is kind of great resource for us and everybody else who's interested in doing this. But we also understand that making the transgenic line takes time. So in the meantime, we also have a transient version of the system. We're using piggyback transposition to integrate both the base editor and gRNA and also barcodes into the cells. That allows you to do a transient experiment. For example, if you have a retina that you electroporate these plasmids in, and I'm just showing here the distribution of number of integrations that we got. So on average, about 3 or 4 integrations per cell. That gives you enough memory capacity to reconstruct few cell generations. So again, what we want to do with this technology, at least for me, is to reconstruct lineage trees and look at their variability in development. Specifically, I guess we're kind of late on the time, but if I want to continue with this, or I want to just question at this point.
Please go ahead, Amjad.
Okay. Yeah. So this, whenever we talk about lineage trees and variability, we have to mention the exceptional case of C. elegans, where you have the same exact lineage tree between different embryos. But this is exceptional. In modern animal models of that, there's a lot of variability in the lineage tree. So for example, here the early divisions is in the mouse, and you can see, and the cell fates are coded by blue and red, and you can see that for 9 different embryos or trace here, very little is in terms of patterns of cell fates assignment of the lineage trees. And this raises a lot of important questions. For example, how much of the cell fate is dictated by its lineage, or if there are patterns that are conserved beyond this variability, that we can basically learn something about cell fate specification from. And in the case of retina that I'm interested in, very young lineage tracing experiments show that progenitors are multipotent, so you can have different cell timing from the same progenitor. But what has been a much more difficult question to answer, that if all the progenitors are basically in their potential, so each one of them has the potential of making all the different cell types, it's just a stochastic effects that make the different lineage trees look different, or if there are patterns or pre-programmed progenitors in the retina that are programmed to make cell types in a specific order or in a specific combination. And some of the insight into answering this question came out from reconstructing the most basic lineage tree that you can reconstruct, which is just labeling a terminally dividing cell, let it divide and have the two cell sister cells that it produces into each other. So these are like basically a doublet tree, and you doing on a specific cells, for example, as that express OLIC2, be able to show that there are in fact progenitors, at least some population of progenitor cells that have this developmental program to make, for example, cone and horizontal cells specifically, which kind of is evidence for model 2. But being able to do this at a higher level, going for example from triplets to quartet, more complicated in the lineage tree, has not been possible because of the limitations of being able to reconstruct trees in this context where at least in vivo you really do time-lapse microscopy. So this is where we want to apply this memoir technology to reconstruct the lineage trees for patterns between them. And the way we basically want to look for patterns is inspired by this work from network motifs, where Uri Alan and colleagues a number of years ago, they were looking at the interaction transcription factors, or even neurons in C. elegans, and to find if there are specific patterns interactions over represented or underrepresented compared to what you expect by random chance. So with this framework of network motifs, where you show interactions randomly and then ask whether there are specific of interactions that are more observed in the real data compared to the mice data. So we basically, inspiration from this work, applied the same kind of logic to the lineages on a preliminary set on the lineages in the retina, and we can already kind of see that beyond those that people have been able to reconstruct by lineage tracing of the differentiating cells. In the triplet also, you can find some interesting that are highly significant compared to what you expect by random chance. And some of them are kind of, for example, you have an amicron and a bipicell with a bipolar as an output. It doesn't really, based on the current our current knowledge, really makes sense why it should be highly over represented in the trees of the retina, but that's the way it is. So this is another kind of aspect that I'm excited to follow, both in terms of molecular underpinning of how this lineage programs occur, also play why they're there basically, and also kind of expanding and making this framework of lineage trees available to the community, because by the advent of more quantitative methods for reconstructing lineage trees at scale, ways of realizing that and making sense of it more and more in demand. So with that, I just want to summarize entire picture here. I can kind of classify it into three class technologies that we can use. One first aspect that I talked about is basically having viral barcoding that has high diversity, allows you to label cells densely and then read those barcodes situ. So it enables you to look at the lineage at the clonal composition of the cells at high density. The type of technology is editable or dynamic barcodes that can accumulate edits over time stochastically, and you can use them for lineage trees of the cells, and then try to make sense of patterns that are over or under expressed, over or under represented in these lineage trees. And then the third type of technology is very exciting, but it's still in more the less mature state of technology is using kind of a sandwich as linear, but now with a reporter for a specific signal to record the intensity of the signal, the order of signals in the gene cells. This is also potential for using the same barcode, the same basically infrastructure for this one as well, but it's less developed at this point. So with that, I will thank everybody in the Elowitz lab, and also our peers in Kai lab and Lewis lab, especially Martin Tran, the graduate student in Elowitz lab, worked very closely with, and Luis Sanchez is a postdoc in Carlos Lewis's lab. So the funding, and again, you guys for taking listening, and also not complaining about all the difficulties.
Great. Thanks, Amjad. That was absolutely outstanding. Really fantastic stuff. So if people want to put questions in the chat, I can either try to unmute you or read them out. I'll start. I have a question. Well, it's question comment, I guess. So I mean, I was really struck by what you said about this kind of stochasticity to the individual steps during development, and yet obviously the typically very robust, consistent output at the end of it. I think that's something that we've been very interested in the context of aging specifically. Okay, so we've wondered whether or not there can be perhaps deficiencies in some of those stochastic developmental processes, maybe due to kind of in utero environmental inputs, followed by maybe some type of homeostatic mechanism which seemingly compensates for those deficiencies, but that comes at a cost in later life and may actually lead to unhealthy aging or decreased longevity in later life. So I'm not a developmental biologist at all, but I'm just wondering whether or not there's any evidence for those kinds of homeostatic mechanisms that are built into the developmental process, which might be operating in that type of way.
That's an interesting question. But so the point is that the question of variety, especially for anything besides in vitro systems, bacteria or yeast, it is very understudied because of the limitations. So we don't know much about it, and it's usually thought of as maybe you thought of it as something to be dealt with. So you have a system that you want to work robustly, but you have this variability, so you have to somehow around it or be able to overcome it. But there are also situations where this variability can be useful. At least in the case of bacteria and yeast, you can find situations where organisms advantage of this stochasticity or this variability to hedge their bets or make sure that overall the population survives. So there are examples of it being taken advantage of in context. But for development, usually people think of it as something that needs to be common, needs to in spite of this, the variable need to be able to make a robust outcome. So I don't think I have a great answer for your question. Maybe someone else does, maybe no one does. But yeah. So about it is that with this high throughput, high densities, we're kind of beginning to start to study that process in the context of development, whole context like regeneration, disease.
Okay, yeah, I mean, hopefully, I've not come across really an answer to the question. So, hopefully, I guess with these technologies, these are some of the questions which you'll be able to start to address, you and others using this technology. There's a question here from Pallav Kasuri. I'm going to see if I can unmute him. Not sure if that's going to work or not. Pallav, can you hear me? Yeah, go for it.
Okay, great. Yeah, beautiful work. I think this is absolutely stunning. I'm really excited to see where you're taking this technology. And I'm curious in terms of the expression of these barcodes, both in cell lines and also in tissue. How much does your ability to accurately and sort of completely read out these barcodes depend on how well they express? Mm-hmm. And is the expression level noise any concern at all in any of your system? Is the natural sort of variation of expression something that causes problems when you try to read them out?
Yeah, that's a really good question. I kind of was over a lot of details in terms of characterizing the efficiency of transcription, both in types and in tissue. But the point is that the expression is not 100%, as you might have expected. It is high enough to be able to do this tricks that I mentioned. It is in the culture something around 85% of the targets being expressed. But one of the interesting things that we found is that each promoter by itself fires independently of the other promoters in the cell. What that means for us is that the reason that we have 80% of the sites expressed is not because for 15% of them there was a barrier for the enzyme to get it to them, or like a fixation or something like that. It is most probably just the stochastic effect that 85% promoters at each individual moment of time are firing, and then 15% of are not. And you can, knowing that, you can kind of take advantage and have redundancies built into the system. So if it is important for your application, so far for us it has not been critical. Just showed it as some fact in the paper. We'll use it later on designs. But if it is very important for you to have a higher level of detection or expression, you make redundancies in your system where you have multiple motors of the same type drive the expression of the barcode. And then because each one of them is firing independently of the other ones, you can reduce that percent not expressed barcodes to, for example, 2%. This is something that, again, we kind of showed just in cell culture in the paper, but this is not become an important issue for us to incorporate it in all the design stages later on. Sure.
Okay. I think you've dazzled everybody, Amjad. There's not that many questions coming through. I have another one though. So you know, I think. Sorry, did someone say? Did someone? No. Okay. So I think a really exciting application of these kinds of lineage tracing technologies in mouse is obviously to look at the clonal evolution of cancer. But I'm wondering if you've given much thought to the cancer models that you might apply it to, because as I see it, one of the weaknesses of many cancer models at the moment is that a lot of the commonly used models don't actually depend upon clonal expansion. Okay, so for example, the KPC mouse model is a mouse model where the mice have activated RAS and inactivation of P53 in all of the cells of the pancreas, pretty much from birth. So not so relevant for studying, I think, clonal expansion from a single mutated cell. So do you have any thoughts as to what cancer models you might apply this to, or how this might lead to perhaps a rethinking of some of the mouse models that are commonly used in the field?
Yeah. First of all, I think really. So I try to for my own research, because of my background in development, my interest. I'm kind of trying to use this technology in a developmental setting, but there's nothing really spatial about it that's more suitable for development to cancer or other applications. So they are relatively general. And also, so really the impact of this type of work is maximized where you have collaborations, or you have other teams using it applied in a lot of different systems. So we are really interested in kind of having collaborators that can approach systems, even though we are not cancer experts. I think that a lot of models that well, my knowledge in this area is very shaky, but I think a lot have been developed so far. They've been built for the technologies that were present at the time. So having a continuous population of cell transform is an advantage, where you can't really quantify the variability, or where the variability gonna show as a bigger error on your plot, basically. So having the ability to trace individual cells or hundreds of individual cells simultaneously is going to enable to answer different types of questions, also require different types of models for those. What exact models are going to be, I'm not really sure, because I don't know as much about like making cancer models.
Okay. Yeah, I agree. I think might need some more sophisticated mouse models to really make the best of this technology.
Yeah, if you're making a homogeneous mutation of the cells, you might expect similar effects happening. Yeah, you can also, I mean, if there's when patient sample can culture and expand in the lab, if you have access to that type of samples, you can also imagine transduce those samples and then culturing them, then tracing the lineage. So that might be more realistic. But for mouse models, I'm not really sure what.
Right.
Yep.
Okay. Well, if there's no more questions, I think we'll call it a day.
Thank you so much.
Thanks very much, Amjad. It's really beautiful work, and I'm sure we'll be seeing a lot more of it in the future. Thanks.
Sure. Yeah. Have a good day, everyone. Bye.
Bye.